for coming. My name is Reedy. I'm a sixth-year grad student in Bob Cox's lab at UVA, and today I'll be sharing some data from my ongoing dissertation work. My work over the last few years has been focused on interlocus sexual conflict, and that conflict is illustrated here. You can see there are phenotypic differences between the sexes. In this example trait, we have plumage color. Males are brighter, females are drab. But neither sex is centered below their sex-specific phenotypic optima. So selection is pulling the sexes in two different directions and generating this sexual antagonism. This idea for genomic sexual conflict has been around for quite some time. It was described in the 70s by the likes of Trivers and Dawkins, and most notably Jeff Parker in a 1979 book chapter. But despite being a pretty well thought out idea, it wasn't until decades later that empirical work really got going on this topic. And one of the earliest, and still one of the best examples, came to us from this paper by Chippendale, Gibson, and Rice in the early 2000s. And what you see here on the left, this is data from the juvenile life stage in Drosophila. And because it's Drosophila, you can do all sorts of cool things. Each data point is a hemiclonal lineage, and they're able to put that genetic background in a male or a female. And what you see from the data here is that the genetic packages that confer high fitness for males, so male fitness is here on the y-axis, are the same genotypes that confer high fitness for females. So you see this clear sexual concordance at the juvenile life stage when viability is the measure of fitness. But then you take these same genotypes and you look at adult fitness, and you're looking at reproductive success here on the right, and you see that the genotypes that confer high fitness in males confer low fitness in females. So this is completely reversed. You no longer see concordance, but you see this sexual antagonism in the adult life stage. So this leads to this hypothesis that sexual antagonism increases over ontogeny. And despite this sort of early work that I showed, showing this conflict developing over the life stage and developing in the adult stage, a lot of the sort of more modern work on sexual conflict actually hasn't tested this. So I wanted to test this in my study species, the brown anole. And I think brown anoles are a good system to test this for a couple of reasons. If you look here on the left, these are juveniles, males and females. Females bordered in red, males bordered in blue, and you really can hardly tell the difference between them. They're almost completely monomorphic. As they become adults, they get very pronounced of sexual dimorphism, where females are much smaller than males. Males are about 30% longer. They're about 3 times as heavy. Males also have this greatly enlarged dewlap, which is an order of magnitude bigger than the female dewlap. So my idea here is that you'll see sexual concordance in the juvenile life stage, and as you move to the adult life stage, selection is going to be acting differently on the sexes. Another reason this species is really good to test this sort of hypothesis is they're very amenable to measuring selection in the wild. They occur at incredibly high densities. They're fairly easy to capture and mark, so they're a good study system to test this in the wild. And these are these figures illustrate the general predictions of the hypothesis here. On the left-hand panel, you see this is the juvenile life stage, and you see this prediction of sexually concordant selection. The relationship between a trait and fitness is going to be the same for both sexes. In the right-hand panel here, you see the prediction for the adult life stage, where I'm predicting sexually antagonistic selection, where the selection is going to be acting very differently on the same trait in males and females. So my work has taken place on a series of spoil islands in Florida's Intracoastal Waterway. This is Island H, where the bulk of my dissertation work has been done. A lot of the data from this talk will be from there. And I'm also going to pull in some data from another island just a little bit south of the Island H. This is in northern Florida. So zooming in, this is Island H. And it's really the perfect size to do this kind of work. It's got a radius of about 96 feet. To give you a better idea of the size, if you were so inclined, you could fit a baseball field on the island. Fit perfectly. No outfield, but the infield fits there. We sample lizards on this island 4 times a year. And when we go, we're doing near complete population sampling. Program Mark estimates that we get 90 to 95% of the animals captured on any given trip. And we're there 4 times a year: March, May, July, October. You can see sample sizes on the left. This island holds an incredible number of lizards. If you haven't worked with brown and holes before, you may be surprised to just sort of visually see how many lizards can be held on an island this size. And so these dots represent capture from one sampling effort in 2015. They are at incredible densities. The population was close to 1,000 adults at this point. You also might notice that population numbers are a little bit down for the in progress in 2017, but that's because the island was raked by Hurricane Matthew. So I'm going to give you an example of the kind of data that we're collecting with each individual lizard capture. So here in this example, this is lizard 4503 male, and he has a unique toe clip identification, so we know who it is when we recapture. And we're measuring this suite of traits. We're measuring snout vent length, dewlap area, head length, head width, and hind limb length. And we're measuring that for each adult lizard that we capture. When we return, if we capture the animal again, it becomes a 1 in the data set as a live. If it's not there, it's a die and it's a 0. If we do happen to miss an animal and catch it on a following trip, the database gets amended. And we now have phenotypic data, we have survival data, so we're using that to get selection gradients. And so you can kind of see here with raw values, on the y-axis you have the live or the die, and on the x-axis you have the trait value, just raw value in millimeters. To convert that to a standardized linear selection gradient, you take that and you convert, so now the y-axis is relative survival, so this is just 1 or 0 divided by the population mean survival. And then on the x-axis, we're taking those raw measurements and converting them to a standardized measurement with a 0 mean and converting it into units of standard deviation. And then the slope of that line, this gives us the selection gradient or beta. In this example, I've given you just a kind of a simplistic univariate case where it's a selection differential. When we're actually looking at all 5 of those traits, that beta is a partial correlation coefficient from a model with everything in it. For adults, we're measuring all those traits. For juveniles, we're only measuring snout vent length. And so all the comparisons I'm going to make between adult and juveniles are through this lens of this single trait, which is body size. So here's some results. This is selection on juveniles for 2 years, and this is from the interval of July, which is the first sample in a season where hatchlings appear, and survive into October. And what we see in both years, selection is a little bit different from one year to the next, but selection is acting the same way on this trait of body size in males and females. So this fits very well with the prediction of sexual concordance at this life stage. Let's take a look at some of the results from adult selection. So when I look at adults, you immediately see a different pattern with the eyeball. You see sex differences in selection at the adult life stage. So this fit very well with the general predictions. A couple of subtle differences, or a subtle difference I want to point out here. In 2015, you have essentially no selection on female body size, and you have positive selection on male body size. So this might not be thought of as actual antagonism, but it's a clear sex difference in selection. And then in 2016, you see the selection on males was reversed. In 2016, it was better to be a smaller male in terms of your survival probability. But what you see here is selection acting in 2 different directions. This is more of actual antagonism. So to test the hypothesis more rigorously, I would need more estimates of selection than just these 4. And I would also need some way to sort of quantify how antagonistic is the selection. So in order to do that, first to sort of get a bigger sample size of selection gradients, what I've done is now, instead of just looking across a whole season, which is what I was doing for the adults in that last slide, now I'm measuring selection over smaller intervals. So I'm measuring from March to May, May to July, July to October, and then in the following year, the same schedule. So that gives me 6 estimates of selection for adults over there. And then for the juveniles, this is the light purple, just this July to October interval, and then 1 in each year, so that's 2. And then I'm also pulling in data from this other island that I worked on previously. The sampling schedule was a little bit different, but it gives me 3 more estimates of selection on adults and 2 more on juveniles. So now I have a few more data points to do this. And then to quantify how antagonistic the selection was, I used this joint index for the intensity of sex-specific selection. This came from Innocenti and Morrow 2010 paper. And the way this index works, you can see the equation there. You're putting in the selection estimates from both sexes. But if you have no selection in one sex, no matter what selection is doing in the other, you're always going to get a 0 value. If you have selection going in the same direction in both sexes, whether that's positive or negative, you're always going to get a positive value of the index. And then it's only when you have selection going in opposite directions in males and females that you get a negative number indicating antagonism. So results, when I look at this all together, the first thing that jumps out here, so this is selection on juvenile size was strongly concordant. Although those 4 estimates of antagonism in juveniles gives us a very positive value. And in adults, I don't see antagonism, but what I see is neither antagonism nor concordance. Most of the values of that index end up being right around 0. There are a couple of point estimates that dip into negative territory, but they're around 0. If you work with anoles, you're familiar with juvenile anoles, this result also can sort of prompt a question about this result. I'm going to expand this and look at it in a different way. One of the problems with working with anoles is that there's enormous age structure in the population of juveniles because female anoles lay a single egg roughly every week from May to October. So when we go and sample thousands of juveniles at one time, there's a bunch of different ages in that sample. So what I did is I took the biggest hatchlings, over 30 millimeters, and I threw them out of the sample. They're approaching adult size. And then I partitioned the remaining into 2 size classes. Hatchlings, which are 23 millimeters or smaller, that's basically the size they come out of the egg at. And then juveniles, which are 24 to 30 millimeters. And here, what I still see positive concordant selection in juveniles, in hatchlings. So I think it is bigger is better. It's not just all age variation. But interestingly, in the juvenile stage, we get a lot of antagonistic estimates of selection in that sort of intermediate juvenile size class. It could be that at this point, selection is acting very differently on males and females, but there's very little sexual dimorphism. So that's an interesting result that I'm looking to add to this year with more data. So I also have data from all the other traits for adults, and this can't directly make a comparison between antagonism in the juvenile and the adult stage, but it can potentially tell us something about the frequency with which sexual antagonism occurs. So when I look at all 5 traits, and so this is 25 individual estimates of selection, so 5 different traits over 5 intervals, what I see is that sexual concordance and sexual antagonism occur at just about the same frequency. You just get this spread of estimates centered around 0 on that antagonism index. When that